Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about transcription. Transcription is a biological process in which RNA is synthesized from DNA with the help of enzyme DNA-dependent RNA polymerase. The term transcription unit refers to the DNA segment that is transcribed to produce an RNA molecule. In prokaryotes, DNA-dependent RNA polymerase consists of 4 distinct subunits. The 4 distinct subunits are alpha subunit, beta subunit, beta prime subunit, and another sigma subunit. So these 4 distinct subunits together constitute the holoenzyme. The holoenzyme of RNA polymerase is composed of 2 alpha subunit, 1 beta subunit, 1 beta prime subunit, and another sigma factor. This holoenzyme is necessary for initiation of transcription. After initiation of transcription, the sigma factor is dissociated from this holoenzyme. After that, the process of elongation can be carried out with the help of core enzyme. So, the tetrameric core of RNA polymerase is composed of 2 alpha subunit, 1 beta subunit, and 1 beta prime subunit. The molecular weight of prokaryotic RNA polymerase is about 465 kilodalton. The alpha subunit is required for assembly of the tetrameric core, whereas the beta subunit, it possesses the ribonucleoside triphosphate binding site, and the beta prime subunit, it contains DNA template binding region. In transcription, RNA is synthesized from a DNA template. In a duplex DNA, only one strand serves as the template strand. The template strand is also known as antisense strand or non-coding strand. And the process of transcription always proceeds in 5' to 3' direction. The base sequence of the growing RNA chain is determined by the sequence of this template strand. The remaining strand is known as coding strand or sense strand or non-template strand. So in a double-stranded DNA molecule, only one strand serves as the template strand. For the process of transcription, the enzyme RNA polymerase must bind to the DNA. The enzyme RNA polymerase binds at some specific site on the DNA. That specific region at which the enzyme RNA polymerase binds is known as promoter sequence. In E. coli, the promoter contains 2 conserved sequences. One conserved sequence is located at a distance of 10 base pair upstream from the starting point of transcription. This is the starting point of transcription. So from this starting point, at a distance of 10 base pair upstream, a sequence is present that is rich in 80. So this 80 rich sequence is also known as -10 sequence. This sequence is also known as Tata box or Prigno box. Another sequence is located further upstream at a distance of 35 base pair. The sequence is known as -35 sequence. The consensus sequence is TTGACA, whereas the consensus sequence of Tata box is TATAAT. So these two conserved sequences play a very important role in the initiation of transcription. The enzyme DNA dependent RNA polymerase first bind with this -35 sequence. After binding at this -35 sequence, localized unwinding occur. Localized unwinding occur at this AT rich sequence. So the localized unwinding that occur at this AT rich sequence leads to the creation of transcription bubble. So the transcription bubble is a specific region in which there is localized unwinding. After localized unwinding, the process of initiation starts. The distance between -35 sequence and this -10 sequence is highly conserved in E. coli. The distance do not exceed 20 base pair and it is not less than 10 base pair. And the initiation of transcription begin at a distance of 5 to 9 base pair beyond the AT rich sequence. Friend, this illustration clearly describes the process of initiation of transcription. The process of transcription involves 3 stages: initiation, elongation and termination. First of all, we shall discuss about initiation. The initiation of transcription involves 3 steps. First step is the binding of RNA polymerase holoenzyme to the promoter. Second step is localized unwinding of the AT rich sequence that leads to the formation of transcription bubble. Third step is the formation of first few phosphodiester bond between the incoming ribonucleotides of the nascent RNA chain. In this illustration, this is the -35 sequence and the enzyme RNA polymerase bind with this -35 sequence, and this is the AT rich sequence. This is the AT rich sequence. So the binding of RNA polymerase to this -35 sequence causes unwinding in this AT rich sequence. So there is localized unwinding. So this transcription bubble has been formed. The process of transcription is initiated at a distance of 5 to 9 base pair beyond the AT rich sequence. So friends, this is the transcription start site. In this double-stranded DNA, this strand running 3' to 5' direction, and this is the complementary strand that running 5' to 3' direction. This is the non-template strand or coding strand, whereas this strand is the template strand or antisense strand. The base sequence of the RNA chain will be decided by the sequence of this template strand, and the transcription, as you know, always proceed in 5' to 3' direction. During initiation, the formation of first few phosphodiester bond formation occur between the incoming ribonucleotides. The RNA polymerase holoenzyme remain bound with the DNA during the formation of 8 or 9 phosphodiester bond. After that, the sigma factor, which is a component of the holoenzyme, is dissociated and the process of elongation will be carried out by the core enzyme. After initiation, the next stage is elongation. In E. coli, in 90% of the cases, the 5' nitrogenous base of the growing RNA chain is a purine. The process of elongation is catalyzed by RNA polymerase core enzyme after dissociation of the sigma factor. After dissociation of the sigma factor, RNA polymerase core enzyme extend the RNA chain processively by adding ribonucleotides to the 3' end of the growing RNA chain. The process of elongation is carried out within transcription bubble. In the transcription bubble, there is localized unwinding. In E. coli, the average length of the transcription bubble is 18 base pair, and this is the enzyme RNA polymerase core enzyme, and this is the transcription bubble. Elongation takes place within the transcription bubble. The enzyme RNA polymerase possesses two activities. One is unwinding activity and rewinding activity. During movement along the DNA, the enzyme RNA polymerase unwind the DNA double helix that is ahead of the polymerization point. At the same time, the enzyme also rewind the complementary strands that are present behind the polymerization point. During the process of elongation, this nascent RNA chain, this blue colored strand is the growing RNA chain. During elongation, this growing RNA chain is displaced from the template strand. So the process of elongation can continue. The transient base pairing between the nascent RNA chain and the template strand is only about 3 nucleotides long. The stability of the transcription bubble is maintained by the binding of the enzyme RNA polymerase with the template strand and also with the growing RNA chain. This is about elongation. The last stage of transcription is termination. Termination occurs when the enzyme RNA polymerase encounter termination signal. When this encounter occurs, the transcription complex dissociates and the enzyme RNA polymerase is separated from the DNA. At the same time, the growing RNA chain is also released. In E. coli, termination of transcription occurs by two different mechanisms. One is row-dependent termination, other is row-independent termination. First, we will discuss about row-independent termination. The row-independent terminator contains G-series sequence. This is a sequence which is G-series. Similarly, these sequences are also G-series. These G-series sequences are inverted repeats. What is inverted repeat? Now, these two sequences, if read in opposite direction, it will be same. After inverted repeat, there are 6 or more adenine thymine base pairs in which the adenine residues are present on the template strand. After transcription of the inverted repeats, due to base complementarity, formation of hairpin-like structure occur. This is the hairpin-like structure, part of which is double-stranded. Due to the formation of this hairpin-like structure, the enzyme RNA polymerase failed to move further because the enzyme RNA polymerase is retarded due to the formation of this hairpin loop. In addition to this, the base pairing between uracil and adenine is more weak as compared to other base pair. So because of the formation of hairpin loop and also because the base pairing between uracil and adenine is weak, so the growing RNA chain is easily separated from the DNA. So friends, this is the hairpin loop. This hairpin loop has been formed due to the base complementarity, and in the last step, this is the hairpin that has been released. So due to this, termination occurs. Next, row dependent terminator. The mechanism of row dependent termination is same. In row dependent termination also, there is formation of a hairpin-like structure due to the presence of infected repeats in the DNA template. In case of row dependent termination, there are two additional sequences. These two additional sequences are present upstream of the G-series sequence. One sequence contains more guanine residue but few cytosine residue. Due to this, after transcription, hairpin-like structures are not formed. Next, there is another sequence that specifies a binding site for the protein known as Rho factor. Rho factor is a protein that binds to a specific site known as root site. After binding of the Rho factor to the root site, termination of transcription occurs because due to the formation of hairpin loop, the enzyme RNA polymerase failed to move. But the Rho factor, it can move further, and because the Rho factor is a helicase, it can easily separate the growing RNA chain from the DNA, thereby causing termination of transcription. So friends, this is about transcription. So friends, please watch and like this video and also share it with your friends. Please subscribe my YouTube channel, Sam Online Class. The subscription of my YouTube channel is absolutely free of cost. Not a single pie is required for subscription. Thank you very much friends. See you. Bye bye. Good night.